Welcome to Beekeeping Today podcast presented by Bee Culture, the magazine of American beekeeping. Beekeeping Today podcast is your source for beekeeping news, information, and entertainment. I'm Jeff Ott.
And I'm Kim Flott, I'm editor of Bee Culture magazine.
Today's podcast is sponsored by Dr. Larry Connor's Wickwasp Press. Dr. Connor has published over a dozen titles dealing with bees, beekeeping, queen rearing, and pollination. At the Wickwasp Press site, you'll find a great selection of must-have books for beekeepers and people who love honeybees, such as Foraging Afar by William Bloomquist, Beecabulary Essentials by Andrew Connor, Garden Plants for Honeybees by Peter Lintner, Increase Essentials and Queen Rearing Essentials by Dr. Connor, and Swarm Essentials by Steve Rapasky with Dr. Connor. All of these and more are at www.wickwasp.com. That's www.wicwas.com. And while you're there, be sure to thank Dr. Connor for sponsoring the Beekeeping Today podcast.
Hey, Kim, I'm looking forward to today's podcast. Dr. Ramsey's done some important work, and we kind of opened everyone's eyes.
Yeah, as you know, Jeff, he's just finished his graduate work with Dennis vanEngelsdorp at the University of Maryland, and now he's moved on to being a researcher at the USDA Beltsville Bee Lab with Jay Evans and the crew there. And I got to tell you, once you listen to what he has to say and you get a chance to absorb it, I think this is a game changer when it comes to dealing with Varroa.
I've watched his YouTube videos, all of his YouTube videos, and it's pretty amazing. So let's call him up on Zoom and get right into it. Well, great. We have now with us Dr. Samuel Ramsey, and it's been a great opportunity to get Dr. Ramsey on the phone with us and talk to us about his latest research and everything else that's going on in his life. Thanks for being here.
Of course. It's a pleasure.
Just a quick question, Dr. Ramsey. Where are you right now, and who do you work for? So we got a good feel for your background and your present.
For sure. So I work for the Honeybee Research Lab. It's a part of the United States Department of Agriculture, so one of their departments. And my work is, at this point, really just focusing on figuring out how to weaponize the things that I discovered during my PhD. So now that we know what the Varroa mite is eating, now it's time to figure out how to use that to kill it.
About as succinct as you can get, but that's why we're here today. What have you found relative to what you just said? That's the background that people are interested in.
Oh, for sure. It's funny that I've kind of started to get notoriety based off of this fat body discovery, because people have been referring to me as the fat body guy. Should you ever meet me in person, you'll find that is not the most apt description of me, but well, we'll go with it. I like it. So I've become the fat body guy, and specifically because there is, for a very long time now, for several decades, it's been said that the parasite, the most damaging parasite that can be found in honeybee colonies right now, it's been said that it's feeding on the bee's blood. And my work has actually found that it's feeding on a specific organ called the fat body. The fat body is different from just body fat. I hear a lot of people try to summarize things and say, Dr. Ramsey's work has shown that Varroa destructor feed on body fat and not blood. But it's actually very different from that, because the fat body, while it does encompass most of the body fat of the bee, it is an organ that has 9 essential functions that it conducts inside of the bees to keep them healthy, everything from its involvement with immune function to pesticide detoxification. It balances and regulates hormones. It creates a number of different proteins that the bees need. It's actually the primary place where proteins are made in the bee's body. It does a lot of important stuff. So
that's analogous to a human liver then.
Yeah, it's very similar to the human liver in a number of different ways. There's also some functions that it conducts that aren't analogous to the human liver, because there's just things that bees need to do that we don't do. But it's a very important set of tissue. It's a very important organ, and I think people are really starting to pick up on that more now than they have in the past.
Well, I know there's some of the people you worked on this project have some history with this. Can you go back a little bit and give us, a lot of people didn't pick up on this, and the question isn't why, but how did you guys pick up on it?
For sure. So I love getting that question because it's important to me to be able to let people know that this did not happen in a vacuum, but there were a number of people involved in this process. And so Alan Cohen, I'm not sure exactly when, but Alan Cohen, he's one of the researchers who was on the seminal paper describing the excrement produced by Varroa. And it's weird to talk about excrement when you're talking about insects, but you can learn a lot about a creature from what it excretes. And so they described the excrement of this organism, and it wasn't the sort of excrement that you would typically expect from a creature that's feeding on blood, but specifically on blood that's so dilute in its nutrient content. And so that seems to have been something that Dr. Cohen thought was pretty strange enough for it to bother him. And he talked about it with a couple of researchers. I know he mentioned it to Jamie Ellis, and he mentioned it to Dennis vanEngelsdorp, and just saying, there might be more to this story than these mites feeding on the bee's blood. They're probably feeding on something else entirely. And so I know Dennis had been processing this for a while. Dr. Ellis had been processing this for a while. And so when I joined Dennis's lab, he threw out this idea to me. Well, maybe you could, since you're so interested in parasites and symbiosis and relationships between organisms, and since you've got experience looking at parasites, why don't you look at this Varroa mite and see if maybe there's something interesting going on with the feeding. And so it was kind of a big shot in the dark there, because there's a lot of options for tissue. There's a lot of things going on inside of a bee's body. But the more I started to dig through the layers of what was going on, starting from the excrement and working all the way through all the other things that came up as a result of studying that, it eventually led to a bunch of experiments that brought us to a very clear conclusion that they're feeding on the fat body of the bees.
And that's of the larva as well, right? Because all the current literature, or up until this year, said that it primarily fed on the larva and bred underneath the cap. And so everything was happening there. So what made you look at what was actually going on the bee as opposed to looking at the larva?
See, now this is the part where my inexperience with honeybees actually served me. So I've been an entomologist for pretty much ever. I've been interested in studying insects since I was 7. I told my parents when I was a 7-year-old that I was going to be an entomologist when I grew up. So I've had a lot of interest in several different kinds of insects, but I've never been a beekeeper. I've never spent an extensive amount of time studying bees specifically. And so when I jumped into this, I felt totally inadequate, because honeybees are extremely complicated. Their societies are complicated. Their body plans are interesting and really, really complex. And so I wanted to know everything there was to know about the dynamics as far as the honeybees go, as well as the parasite. And so I did a lot of research on the biology of Varroa. I wanted to know everything from the start. So I went back to the first paper where Varroa was described in 1904, and then tried to look at all the papers since then in the most extensive literature search that I could do to understand this creature. And I think the other thing that's really served me in this has been the fact that I look at a lot of these matters from the perspective of the parasite rather than just the host. And so a lot of researchers have tackled this from the direction of looking at it from top down, from the bees down to the mites. But looking at it from the mites up to the bees, it seemed odd that the mites would waste such an important resource as the adult bees and the important tissues that the adult bees have, especially since the mite's body is very well. This was actually described in 1904 by Dr. Eudemons, who described these organisms. But he described that the domed nature and sort of ellipsoid flattened body that they have would be perfect for fitting between the plates that make up the adult bee's body. And it was really interesting looking at that, because when I would collect bees from the colonies, I would find them between these plates. And when I would separate the plates to look at what's in between them, there's this really thin membrane. I ripped it pretty much every time I separated those plates on the bee. There's this really thin membrane that the mites could potentially be feeding through. And when we did more research on the physiology of the bees, directly under that membrane is the fat body tissue. So I wanted to look at how the mites were impacting the adults, because that led us to a really important implication. We have natural brood breaks, but we also periodically create these artificial brood breaks. And that would force the entire population of mites from the larvae onto the adult bees. And if the mites are actually impacting the adult bees, this may actually be more damaging and more risky than we've considered in the past. And it doesn't mean that it's necessarily a bad idea, but we should be considering that risk when we're moving this whole population, because if they are feeding on the adult bees, they're bringing with them all those viruses and all the damage that they cause in the process of feeding. They're moving all of that into the adult population all at one time. Wow. And that could be really damaging at certain times of year. So I wanted to look at that and see if I could verify and determine pretty clearly whether they were feeding on the adult bees or not. A lot of the implication in some of the literature is that they aren't, while other researchers have assumed that, well, they have to be feeding on the adult bees, because if you put them, if you separate them from adult bees for more than a couple of days, they die. If you leave them with adult bees, they survive. And so they're probably deriving some sort of nutrition from the adult bees. But to directly show that is really, really difficult. And so some of my work focused on the adult bees, some of it focused on the larvae, and it was able to bring together a more holistic idea of how the mites are impacting the entire organism.
Wow.
Sorry, was that too much all at once?
That's a good. So the thing that comes up to me right off the top of that is that one of the management techniques that beekeepers routinely recommend is to artificially create a brood break so that you expose mites. You get mites out of there. There's no mites in cells, and they're all exposed on adults, and therefore more likely to come in contact with whatever treatment you're using. If I understand what you just said, that may be the worst thing we can do.
I wouldn't say the worst thing, but I would say that it is definitely more risky than we have considered in the past.
Yes.
And that's not a guess. That is clear based on the work that we've been conducting, because and that comes from an issue that we've had in the past with terminology. When Varroa was first described, it was described in the former Soviet. When several of the fundamentals of Varroa's behavior and biology was described, a lot of that was described in the former Soviet Union. And a lot of the work that was written about them was written in Russian. And so a lot of those papers have been very difficult for us to actually read. And so for a lot of work that was done about Varroa, we would just look at the abstract from those papers, because that was the only part translated into English. And there was never really verification done on the methods or a lot of the work conducted for some of the other work done there. And so Varroa, their life cycle was separated into two phases: the phoretic phase and the reproductive phase. And we understand that in the reproductive phase, reproduction occurs. But the term foracy is a term that people don't hear very frequently, and it's just not common vernacular. But foracy, it comes from the Greek word which means to bear. It means to take something and move it. And the idea of foracy is that a creature is only used as a shuttle, as a means of transportation to move one organism to another place. That creature is not being used as a trophic resource, so it's not being fed on, and it's not being used as a reproductive resource. And so in order for Varroa to actually be phoretic, they cannot feed on adult bees. period. No feeding whatsoever. And so for a long time, because we've been using this term to describe Varroa for so long, I think there's been people have somewhat taken for granted that Varroa actually impact the adult bees. We kind of think of them as simply using the bees as buses to get them from either one colony to another, to get them from one brood cell to another brood cell, or to switch between adult bees. The problem there, of course, is that if they're simply trying to get from one brood cell to the next, they don't need to stay on adult bees for 7 to 14 days, which is the average amount of time that they're staying on adult bees. And if they're trying to get outside of the colony and fly from colony to colony, they should be attaching to foragers. But the literature shows very clearly that they prefer nurse bees. So those were some of the things that led me to believe they're probably feeding on the adult bee populace as well. Through my work, I've been able to show that they do feed, and they feed a substantial amount on adult bees. And so what you said a moment ago is actually very true. There is a substantial risk to us forcing the entire population of mites, especially if this is a colony where there's a lot of mites in the brood. Usually the majority of the population at any one time is going to be in the brood. And so if you force a large number of mites onto the adult bees, and we know that these mites are damaging the tissue that is in charge of the immune system, the tissue that's in charge of pesticide detoxification, the tissue that's in charge of their hormonal balance, which regulates task shifting and all of these other matters, you're creating a lot of chaos in your adult bee populace. And worse still, you're creating a lot of chaos in the most important part of your, arguably the most important part of your populace, because you've got the nurse bees that they're maintaining this huge complement of nutrition for the colony in their fat bodies. They're feeding the babies. They're making sure that the next generation for these bees can then be carried on. And they're walking around. There's attendants that are also feeding the nurse and the queen and making sure that she has all the nutrition that she needs to continue producing enough eggs for the colony. So we've got a lot of viruses that can then easily be transmitted and disseminated throughout the entire adult bee populace as they go about feeding each other, as they have these heavily weakened immune systems. It allows for viruses to travel a lot more quickly now that they have compromised immune systems because of these mites and all of the other issues that we were just detailing that come about here. We would expect that they would get a lot worse when we force these brood breaks and the longer we allow these mites to continue feeding the adult.
Let me bring back the word chaos here, because that's what this is relative to fundamental management of Varroa right now. I can tell you 100 researchers that will give a talk in the next 3 days. This weekend, somebody will say a forced brood break is a good way to control it because it exposes the adult bees to whatever treatment you're doing, or to being hygienically removed, or whatever.
And that is true. It does force the mites to be in greater exposure to the pesticides. And it does force them into a section of their life cycle that is more vulnerable for grooming. But we have thought of it as if when we do risk-benefit analysis, we have to have accurate information. You cannot do a risk-benefit analysis without adequate knowledge of what the risks are and the benefits. We've had a good idea of what the benefits are, and we have not had a good idea of what the risks are. And I think in that way, we've had a probably more flowery idea of how helpful a brood break is than how it actually.
Interesting. Boy.
Wow, interesting is exactly right. Amazing is what that is. Well, then taking another step, Dr. Ramsey, if you were to develop some sort of management program for Varroa in a beehive using this information, where would you go with it? What's at the top of your list? Have you taken it that far? Let me put it that way, because you may not have. I realize that the fundamental work you've done here is pretty amazing. Next step, of course, is the application of this. And
yes.
Do you have some thoughts?
I do have some thoughts about this, and I would like to say right now, there is certainly a lot more thinking that I would like to do about this. My project, I was really excited about it because of all the ways that it allowed me to apply different layers of the knowledge base that I built up in the last 21 years of studying insects. And so it was a really multifaceted project, but the place where it was most lacking was an application. And there are other students that, who were PhD students at the same time as me. Natalie Steinhauer is one name that comes to mind. Also, Kelly Kulhanek. I loved how applicable their projects were. Natalie was studying how the different undertakings that we have as beekeepers, how they actually impact the colony and how we perceive that they will impact the colony, and sort of the disconnect between them. And it was an absolutely fascinating project because she was able to take all of that information from the Bee Informed Partnerships collections, the loss and management surveys, and look at what was actually statistically the most likely to lead to your colonies surviving the winter, surviving the summer, and all of that. So it was really enlightening. I got to say, I was a little bit jealous of how cool and applicable that project was. You guys should totally have her on for a podcast at some point. But anyway,
write it down,
I'm writing it down. It's recorded. We're set. Thank you.
Okay. So your question about what I would recommend. I've had a lot of time to think about it, and there's still a lot more thinking that I'd like to do, but I have a good deal of reticence now about being too quick to recommend brood breaks. I think we need to spend a little bit more time really looking at whether viruses are more heavily distributed in the colony during brood breaks, and really looking at the health metrics of the bees during these times. But really, I think what we should be looking for is a way to impact the population of mites that are actually below the capping. What we do now is we add things like amitraz or formic acid into the colony, and we want to allow for these chemicals to be present in the colony long enough so that the brood that is currently capped, the mites that are leaving that brood will be exposed to the pesticide. And so then you have to leave it in there long enough for that exposure. But I think if we can find a way to expose the mites to pesticides below the capping, that would be wonderful. That would be that would be a great way for us to impact the population of reproductive mites, because then we could have a shorter term exposure with these chemicals, and we could really reduce the population where the greatest number of organisms actually are, the greatest number of the parasites. We know that formic acid can penetrate the capping. We need there to, of course, be greater research on what's the most effective dosage, temperature, humidity, to make sure that the cappings are penetrated, and what level of kill we can expect when penetrating the capping. And there's still work to be done there. But if I were to, and remember, this is still unverified, but if I were to speculate and to think through what I would expect to have a really heavy effect on the population, I would expect it to be during the reproductive phase, because not only is the foundress mite inside of that cell reproducing, but her entire family, the offspring that she produces, they are by far at their most vulnerable stage. They can't do anything for themselves. They can't even feed by themselves. If she doesn't make a hole in that bee for them to feed through, all of them will die because their mouth parts are totally squishy and they're not strong enough to actually pierce through the larva themselves. And so their helplessness, if you can penetrate the capping, even if you were to just kill her off and her eggs still hatch, they wouldn't be able to take care of themselves at that point. And also, they are extremely vulnerable during that stage because of how thin and absorbent their own exoskeleton is. It doesn't have the opportunity to solidify and harden until the very end of their development, until the point where they mature and are ready to leave that cell themselves. So the exposure of pesticides during that point can kill the entire family of mites that's then developing. So I think we really need to find something we really need to refine our methods of impacting the reproductive phase in addition to the phase that's on adult bees, because more of them are in the brood than are on the adult bees at pretty much any time in the year except when there is no brood. In addition to that, one more addendum to that, we need to reduce the amount of chemicals that we're putting into the colony.
Thank you.
So
Good.
Data that has data from the Bee Informed Partnership and data from other research agencies has shown that when we look at the chemicals that the bees are exposed to on a regular basis, there's a lot of concern about the neonicotinoids and all of the other chemicals that are out in the environment, the fungicides, the herbicides. By far their greatest exposure to pesticides, the bees themselves, are the chemicals that we put into the colonies to control the mites. And while those chemicals are being put into the colonies, while they are miticides, they're specifically targeted to the mites, while they are at doses that are sublethal to the bees, these combinations of chemicals, the fungicides, the herbicides, and then the levels of chemical miticides that the bees are being exposed to, we don't actually know what all of those interactions do to a colony. It's very difficult to describe and adequately study all of those interactions all at the same time. We're also bringing all of those chemicals into the environment on a regular basis. And environmentally, it's just not the best way to do things. And while it's important for chemicals to be a part of integrated pest management, we would hope that the direction that we take things in as researchers is one where we can be reducing more and more and more the amount of chemicals that are put into the environment. And so my hope is that we can find a way to introduce something, some sort of means into the feed of the bees that will go to the fat body. And when the mites then feed, they'll be exposed to this. Now, I am hoping that we won't have to rely on a chemical insecticide. The work that I'm working on right now has nothing to do with specifically putting an insecticide into the bees themselves. And my worry about that is that there's been a lot of work on getting a systemic insecticide into bees and having it persist in their blood. And that was back when we thought the mites were feeding on the bees' blood. And that was less of a concern because the blood is renewed throughout the bee's life cycle, the same way that our blood is renewed. However, we don't get new organs every day. Like that's the replacement of tissues in the body. You're not going to just get a new heart over time. It takes a long time for those cells to be replaced. And so the fat body, things that are fat soluble, have the potential to store in the fat body and eventually biomagnify into something that can be really detrimental to the colony. My hope is to work on finding a way to disrupt some sort of process in the mites that the bees don't have. Because the mites and bees are from totally different classes, different orders, their bodies work pretty differently in a number of ways. And if we can target a function that the mites have, that the bees don't have, then we have a system where we can knock out a function that's essential to the mites themselves and still allow the bees to persist and be happy without the addition of lots of different chemicals into the system. Now, I know that was a long answer. Eventually, that's going to be cut up into a much shorter answer. But what I don't want people to hear me saying is that I think chemicals are something that shouldn't be used in colonies and that we should all go as chemically free. I think that chemicals have a place under certain circumstances. But what we forget is that integrated pest management means that you don't just use chemicals. You find some way from better understanding of the organism that you're attempting to impact, you find other ways of impacting this creature, and you only use chemicals when necessary, when things reach a certain threshold, when you desperately need to reduce the population of the organism and other means have failed. And oftentimes chemicals become the first go-to because we know it's really quick. You can stick these strips in the colony, close it up, walk away, problem solved. And so we don't want that to be the thing that we immediately go to because of the ways that it impacts the rest of the environment, the bees, the ways that it can get into our food and enter the human food chain. And so, yeah, think about it.
So you've given it some consideration.
That was a perfect long answer.
It was.
I think no, it was. It brings all of the, or as far as I know, all of the points that need to be addressed here, from no poison to all poison or only poison, and lots of things in between. I am amazed at how much we missed this target over the years. And I was talking to you earlier about working with Dr. Eric Erickson when he was looking at the guanine deposits in cells of the fecal matter of mites. And it got taken then only as far as it was an indication there were mites in your colony because there were fecal deposits there. He was working with the people that you picked up on also eventually, but they only used it as a road sign that said, mites here, as opposed to what is this stuff and what should they be using? So the next step was
it's worth considering that people usually don't like to focus on poop too much. So. Takes a special kind of researcher.
I can't argue with that. Yeah. Well, so what?
And on that note,
you're at the Beltsville lab.
I am. I love being at the Beltsville lab.
Yeah.
Well, I'll give you a heads up, and we're talking with Jay. We're going to be looking at all of the people working at the Beltsville lab here coming down the road. So the show that we're doing, it's called the Kim and Jim Show, and we will be looking at what you're doing. Hopefully you can do some of that. And speaking of looking at, I know that in our next issue of Bee Culture Magazine, you were talking with Ann Harmon. I did a short interview with her, and you sent along some pictures of the mite actually penetrating that membrane you were talking about and feeding on the fat body. You had a cross section, and there you are. That question asked, question answered. That pretty much solves all of the things that you were talking about with that one photo. It was great. So I encourage people to take a look at the December issue so you can see that, but also to check the Kim and Jim Show coming up sometime soon, Jay said, and maybe we can get a little closer look at some of the things that you were doing. Jeff, you've got a little less experience with this than even I do, and I certainly don't have this much experience that Dr. Sam does. What's your opinion of this, just kind of from a less experienced perspective?
Well, excuse me. It's pretty exciting. I have so many different thoughts that want to jumble out of my mouth right now that it's hard to really isolate. There's a couple of them. One that, and this is not to criticize anybody who's currently researching, but are there other Dr. Samuel Ramseys ready to burst through and look at old research and challenge and question it out there? That, I mean, even though, I mean, there seems to be a lot of research out there that could be either reinvestigated or can be continued on, worked on by a new group of researchers. And I think that would be fun to see. And see, I think if they're all like you, then beekeepers are in good hands, as far as I'm concerned.
Well, first of all, I really appreciate that. I also should, I guess I should give a little bit of detail about what goes into a project like that. Mm-hmm. It is risky in a lot of ways to decide that you're going to go back and look at things that were done in the past, because the way that our system for giving degrees, the way that it works is that, well, I shouldn't say giving degrees, earning degrees. When you receive your degree as a doctoral level researcher, what's been shown is that you're able to give an original contribution to the scientific community. We now know something that we did not know before. And it is, when you go back and you look at old work, if you can't see your way to showing something that wasn't already clear from that older work, you're going to have to start over and keep chugging along and with different projects until you find something. And so it can be kind of risky, because these days you don't want to be a career PhD student. All of us have heard the stories of people who have been in graduate school for, a decade or longer, or who have been in a PhD program for a decade or longer, because these days it's not too hard to be in grad school for a decade or longer. But it can be really difficult when you're trying to figure out to find your way in a PhD program, and to better understand how you can contribute something to the scientific community. Now, when I started looking at the subject of Varroa and their feeding, I noticed enough discrepancies where I couldn't stop thinking about it. There were enough odd little things here and there, and there was no single thing. We can't just focus on the subject of the poop. There was no single thing that left me thinking the mites aren't feeding on the bee's blood. There was this confluence of different things. The mites feeding inside of the cell, they are able to produce an egg that is a substantial volume of their own body volume every 30 hours. They're producing this baby that is greater than 30% their own body volume. And to be able to do that every 30 hours is absolutely crazy. But to be able to do that when feeding on something that is pretty much water with a little bit of nutrients sprinkled in there, that just didn't seem to make a lot of sense with what I understood from biology, from digestion, from nutritional analyses. It just didn't seem to make a lot of sense. The excrement that they were producing was one matter, but also the digestive system itself didn't seem like it was structured in a way to accommodate a huge volume of fluid. Their digestive system is just this tube. It's a tube that bifurcates and runs all the way down to the rectum, but it doesn't do what you would expect in an organism that's processing a lot of fluids, because there usually is some place where you are able to exchange all of the excess water out of that diet so that you can concentrate all the nutrients in it. So if you're going to feed on something that is a very, very dilute fluid, the way that aphids feed on plant sap, or the way that certain insects like triatomines, that's a certain type of true bug related to bed bugs and other insects. If they're going to pierce a mammal, and they need to get rid of all the excess water that is in the blood meal that they're going to consume, and then concentrate the nutrients from the blood itself. There's a number of different physiological changes that are necessary for you to accommodate that. And we weren't seeing any of them in Varroa. And so it was those sorts of things where, when I started this, and I think I would recommend this to anybody who wants to look through old literature, I am sure that there is plenty in the old literature. There are plenty of subjects that sound like they're all sewn up, and there's a neat little bow on it. But if you start looking at things and you see multiple discrepancies, start looking into how those things could fit together and what they may be saying about what you're looking at. I did not go looking for a project that would make me a rebel or anything of the sort. Actually, it was quite the opposite. I was trying to find a nice, quiet project and keep my head down. And so when I started this project, I was coming off of a totally different project in a different, working on a different system. I was working on parasites of stink bugs, and I had spent 2.5 years working on that already. And so I only had 3 more years to finish my PhD. And that project, I wasn't able to continue on with that project. I needed a new project that was going to get me all the way through. And so when I started talking about this idea of Varroa dennis, I was still juggling a bunch of other ideas at the same time, and I needed to figure out what to settle on. So I told Dennis, I've got some expectations of these mites, and if these expectations aren't met pretty early on in what I'm able to determine from the literature and maybe some quick experiments on my own, then I might find reason to pursue this project. But right now I'm keeping my options open. And the idea was that I would shut down those other options if I realized along the way, okay, there's more to this story that's been shown. And that's what kept coming up. The things that I just disclosed to you and plenty of other things I kept surfacing. So if you start seeing things that really bother you, that you can't stop thinking about, and they're just sort of floating around in your head, it might be worth pursuing that. But, develop some expectations. Figure out what sorts of things you expect to see in that system and what it might be saying.
That's great advice.
Mm-hmm.
You brought up a subject, and I'm sitting here thinking with a third of my brain thinking something just a little bit different. Where does the jump from Sirena to Mellifera play in here? Because Sirena was drones only. And when they jumped, and there are people who say it was the random change of one gene that allowed Varroa to go from drones only on Sirena to drones and workers on Mellifera. And I have now as much as I know, and I don't know very much at all. But that question, as you were talking about this, came to the top here. And do you have a thought on that? Is that any role at all in this?
I like you, Kim. You've got great questions, I tell you. I love the subject of Apis cerana. If people are looking for other PhD projects or other really important things that need to be studied, it's also a good idea to start looking outside of the U.S. Because if you've noticed, pretty much every 10 years, we get a new parasite of some sort or a new disease of some sort. And it usually comes from some other country. It comes from Southeast Asia, where there are tons and tons and tons of different species of honeybees. It's debatable. Some researchers will tell you there's 11 species of honeybees in Southeast Asia. Some will tell you there's 9, but pretty much they have every species of honeybee. And then Apis mellifera is there as well. And it's that exchange of parasites and diseases and things between all of them that has created this system where we keep seeing these new, what are new to us, but these organisms showing up that can be really damaging to Apis mellifera because they simply don't have an evolutionary history of constantly being exposed to parasites. The bees in Southeast Asia, every species of honeybee that exists in Southeast Asia. And because they've all been hanging out, that's where the parasites hang out. Because the parasites have tons of options of what they're going to feed on and how they're going to feed. And if one of the sets of bees is migrating, they might be able to shift to another set of bees. And so if you really want to study and be proactive about what could be showing up in the future, what's going to be the next big issue, or you want a better understanding of how bees naturally control parasites and disease in the wild, you're going to need to look at some other species of bees because Apis mellifera, there are debates about whether Apis mellifera was originally from Southeast Asia and moved out of Southeast Asia into Europe. And there's lots of debate about this. But what we do know with a good deal of certainty is that Apis mellifera has been separated from the Asian honeybees for a long, long time. And what we know from our understanding of physiology and evolution is that if an organism has some sort of defense against another creature, and it's anything more than a passive defense, if it takes some level of energy or some resources during development, when that parasite is no longer around, what will then be favored is the biology that lacks that particular adaptation. And eventually, the organisms that lack that adaptation will become the dominant version. And so Apis mellifera, lacking all the adaptations to combat these parasites, has become the dominant honeybee because it didn't need those adaptations anymore after it moved out of Southeast Asia and no longer had those parasites.
What that brings up then is, what else are you studying over in that part of the world?
I am studying. So I'm studying the Tropolelaps mite because I am pretty confident. I don't want people to think I've got a crystal ball or anything, or I know what's going to happen in the future. But if we are to learn from history, I have some level of confidence that Tropolelaps is going to be the next introduced parasite into the U.S., for the next introduced parasite of honeybees into the U.S. And the concern there is that we're going in a similar direction that we went in before, where we're kind of waiting until something happens before we do a substantial amount of research about this creature. Originally, it wasn't until Varroa arrived in the country when that country started doing research about this creature. And that's why there's a bunch of early research done in China and in the former USSR about Varroa. That's where all the fundamentals were done, because those are the first two places where it really became a big issue after moving out of Southeast Asia. Now all the work that's being done about Tropolelaps impacting different sets of bees, that's being done in China, and it's being done in Southeast Asia. And if we really want to make sure that we can know as much about these creatures as we could possibly know before they arrive, we really need to start making sure that we're putting energy and resources towards better understanding it. So I have actually been crowdsourcing and raising money to go and study this organism, because it's been more difficult to get this funded through the regular means of funding these sorts of studies. I've heard a few times now that parasite isn't here yet, and we're going to put these resources towards the parasite that actually is here and is impacting our bees. But notice that those individuals were willing to say that organism isn't here yet. There is an understanding that it is on its way, but it's not here yet. And I like the idea of being proactive. So I'm studying this parasite Tropolelaps because it is kind of terrifying. It's about a third the size of Varroa destructor, so it's much more difficult to see. But it is a substantially faster breeder. It breeds more quickly. And part of the reason for that is because it re-enters brood cells much more quickly than Varroa destructor does. While Varroa spends, on average, 7 days on adult bees and as long as 2 weeks, the Tropolelaps mite spends a day or less on adult bees, and usually substantially less than a day, usually around 12 hours or so.
Well, you've opened up an opportunity for the beekeeping community to to make an investment in its future here, I think. And I invite you to tell me right now, how do I do that?
Oh, well, didn't want to do a shameless plug, but since you wanted to throw that in there,
go ahead.
You can actually log on to GoFundMe. And if you type in GoFundMe/fund honeybee research, you will find my page. Also, if you Google Fight the Mite, you will find my page, because it is called the Fight the Mite Initiative. And I am working on figuring out how to fight this mite before it even arrives, because if we have a better understanding of this organism's biology, we might be able to accomplish something that rarely happens when a parasite is introduced. Usually a parasite is introduced and we spend a good 10 to 20 years doing research on it before we have a good idea of what we can do to get rid of it. If the parasite is introduced and we already have ideas about what are effective tools and management measures that can be used against this organism, we can dramatically impact this population and maybe even eradicate it at its point source, rather than allowing it to spread to the entire U.S. before we start figuring out what to do about it. So
we did a good job with that with Varroa.
Yeah, I agree.
And that should be our model of what not to do again, I think. So anyone listening, do something noble here and then spread the word. I think we will be well better off if we start now rather before it gets here.
Well, I appreciate that.
I will share your GoFundMe website in the liner notes here. Liner notes. I'm not dating myself there in the podcast notes.
We actually just reached. So my goal is $25,382, and that will allow me to complete the complement of research in Thailand on this project And that's me trying to figure out what is a reasonable amount and trying to distill that down into the most economical way to get all of this done. And originally I thought that's a huge amount of money. No one's ever going to, through crowdsourcing, be able to actually do that. But through donations, both large and small, but mostly a bunch of small donations, we've raised $16,633 so far, which is wonderful. So there is one final push before March. I need to assemble all of this money before March. So trying to do one last big push. The GoFundMe page doesn't fully reflect that amount. It actually says $9,001 right now. And that is because some people have opted to avoid having GoFundMe take its, I think, 4% or 5% cut off the top. And so they've actually sent those checks directly to the USDA. And so that's gone directly into the account there. But yes, beekeepers can do a lot. I've seen it just by disseminating this to their groups. So thank you very much.
If you would email me the address for if someone wants to contribute directly.
And it's on the GoFundMe page.
Oh, is it? Okay. I'll make sure to include that in the notes of the podcast as well.
Thanks a lot.
Yeah, you bet. All right, Kim, I think we've monopolized enough of Dr. Ramsey's time this afternoon. I could keep going. I'm happy to do that. I got enough disk space here to record for the rest of the week, but I'm sure Dr. Ramsey has better things to do.
Well, I've got to say two more things.
Please.
I am a researcher. I've been a researcher forever, but I'm not. I feel like there is something to be said of having multiple interests. So something that a lot of people have been asking about since EAS. I was actually asked at the Eastern Apiculture Society meeting.
I was there too.
Yeah. I was asked at that meeting to do the banquet, or prior to that meeting to do the banquet. And someone found online that I enjoy singing. And so at the banquet, I just sang a bunch of songs and it was a lot of fun. But I've been surprised for a while now at how surprised people are that researchers do other things aside from research. But if you look at history, some of the most accomplished researchers of any age at any time period were also musicians, were also very interested in several other forms of arts or interested in other disciplines. And so I enjoy having that opportunity to show people how multifaceted researchers can be. There's a lot of fun in that, but I think that sometimes that freeform way of thinking that you can get from certain styles of music can actually help relax your mind and focus you in more creative ways. And I think that has a lot to do with me being more willing to pursue odd ideas in how I pursue my research. I don't think that I'm people have asked before, we missed something big. And so I can't believe we missed this about the mites. And, it's crazy that so many researchers working on this missed something this big. You must be really intelligent to have figured this out. And while I'm not going to downplay that, I know that I'm a hard worker and, I'm a tough cookie. I'm a sharp knife. But I do know, though, that I don't think that I'm any smarter than the other researchers who have tackled these sorts of subjects. But I think that a willingness to look at things differently is really important. And that brings me to my second point. I think diversity is very, very important in science. And so shameless plug here for diversity. There's been a lot of rhetoric going around lately that, there are all these ideas of diversity for diversity's sake, and there's no real value in diversity. We just like it because it sounds good or it makes us feel better or blah, 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 snowflakes. I think I think diversity is really important and can be shown through the work that I've accomplished, because even with the rearing system that I came up with for feeding the mites, it wasn't that system was better than other systems in the past. I just took some I took some odd approaches to being able to feed these organisms. I took some odd approaches to understanding how these creatures impact the adult bees. And I think as long as we continue allowing for science to be a system where the vast majority of people are of the same race, the same gender, the same background, we will have people trying to solve problems the same ways. And I think as we introduce people from different backgrounds, different cultures, I'm not sure if people know this, I am brown. As we introduce people from different cultures and different backgrounds and different ways of thinking, a lot of problems can be solved in different ways than we would have ever imagined that they would be. And so just the plug for diversity there.
I like it. That's very good. I like it. Diversity in all of its forms and factors. I like it a lot.
Absolutely.
All right, Kim.
You share the stage with some other great beekeeper musicians, if you're not aware of C.C. Miller and some of those.
Ah, yes.
Yes. So that's good. Jeff, this has been great. I'm glad that we were able to line this up. The good doctor has spent more than an hour with us, and we appreciate your time. We hope to visit you again on the Kim and Jim show, and you can show some of what you've been talking about here today. What do we got left, Jeff?
I think that's it. I just want to extend the offer that anytime you want to get out your message out to the podcasting crew, that you reach out to Kim and I, and we'd be happy to get you on it at the drop of a at the moment's notice. That's the word I was looking for.
I really appreciate that. It's great to know that I have that opportunity, that open offer available.
You bet. All right. Thanks a lot, Dr. Ramsey.
Okay.
We look forward to
for sure
having you back.
Thank you. Thank you.
Well, that was really something else, Kim. I sat here most of the time just listening to him, not even thinking of what else to ask. He's just a wealth of information and fun to listen to too.
Yeah, the fun to listen to. You know, he has the rare talent of being both a showman and an intellectual scientist, and he's incredibly good at both. And if you haven't yet, certainly tune into his videos on YouTube, because they're entertaining, and you heard him today. You can tell the other half of what he does in his spare time. I think the information, Jeff, that he had today really is, I said it at the beginning, I think it really is a game changer in terms of how we're going to be addressing the Varroa problem from now on. And he's not stopping. He's looking into the future. What I liked about today was what's next, as opposed to how do I fix what's broken now? I'm looking for what's going to break in the future so that I'm ready for it. And to me, that's as good as it gets. And I'm wishing him all of the success. He has, if you recall, he's looking for funding. He's got a fundraiser going, and I think we have the address on there.
Yes, we'll have it up on the website in the show notes.
If it's still going when you hear this. And he's looking for external funding plus USDA funding. So, to me, this was a good day to be here, Jeff.
It was. And I really liked his message about the diversity in terms of bringing all sorts of different approaches to the problems that we are experiencing. So, his unfamiliarity with honeybees and honeybee management and everything else gave him a different perspective on the problem that people have taken for granted, I guess, and that's not a bad mark on anybody else who's been doing all this research for the last 30 years or so. But it just brings a new energy to the problem, and it's fun to see. It's really fun to see.
Yeah, I think that about wraps it up for today, Jeff. I think. Well, we certainly want to thank Larry Connor and Wick-Wasp Press, publisher of dozens of how-to and scientific beekeeping books, for sponsoring this podcast today. You can find out more about Wick-Wasp at www.wickwasp.com. Go out and take a look at their newest publications and give them a high five for helping bring us this great program today.
That's really good, Kim. That's been a great show today, and I think Dr. Ramsey's on a good path. I want to thank all of our listeners and those who've been writing in, and encourage anyone who's listening to rate us 5 stars on iTunes. And that helps other beekeepers find us quicker. And so it's a good thing. So we appreciate the feedback. and keep it coming.
Yep. See you next time, Jeff.
All right, Kim, thanks a lot. Take care.